Navigate to the NCBI BLAST homepage. Under the header, Web BLAST, click on the nucleotide BLAST box. Under Enter Query Sequence, check the box entitled Align 2 or more sequences. Now, you should see two boxes, one entitled Enter Query Sequence and one entitled Enter Subject Sequence. Enter the taster sequence reference number, AY258597, into the Enter Query Sequence box and the non-taster sequence reference number, AF494231, into the Enter Subject Sequence box. At the bottom of the page, select Show Results in a new window, then click on the BLAST button. An alignment analysis will open on a new page. Click MSA Viewer. A graphical representation of your nucleotide sequence alignment will open in a new window. You should see three rows, labeled Consensus, AY258597, and AF494231. As a reminder, AY258597 is the taster sequence, and AF494231 is the non-taster sequence. Above them you will notice a horizontal gray bar with several vertical red lines. These lines are SNVs, single nucleotide variants, or differences in a gene's DNA sequence found within a population. Click the Zoom to Sequence button in order to visualize both the consensus nucleotide sequence and the sequence of the taster and non-taster alleles at each SNV. You will notice that the sequences of the taster and non-taster alleles are mostly represented by dots, indicating that the sequences are identical to the consensus sequence. Only the SNVs are different. What is the position of the first SNV that you find? Move the slider to the next SNV and record its position. You have identified SNVs of the taster and non-taster alleles. Now you will explore whether any of the nucleotide changes lead to changes to the amino acid sequence of either allele. To do so, you will create another alignment in BLAST. This time you will align the amino acid translation of each allele. Navigate to the NCBI BLAST page once more. Under the header Web BLAST, click on the Protein BLAST box. You will be creating a sequence alignment between two protein files from GenBank, the amino acid sequences of a PTC taster allele and a PTC non-taster allele of the TAS2R38 gene. Under Enter Query Sequence, check the box entitled Align 2 or more sequences. Now, you should see two boxes, one entitled Enter Query Sequence and one entitled Enter Subject Sequence. Enter the taster amino acid sequence reference number, AAP14666.1, into the Enter Query Sequence box and the non-taster amino acid sequence reference number, AAM19322.1, into the Enter Subject Sequence box. Select Show Results in a new window, then click on the BLAST button at the bottom of the page. An alignment analysis will open on a new page. Click MSA Viewer. A graphical representation of your amino acid sequence alignment will open in a new window. Click the Zoom to Sequence button in order to visualize the amino acid sequences of the two TAS2R38 alleles in your alignment. You are going to scan the alignment in search of amino acid substitutions. As a reminder, AAP14666.1 is the taster sequence and AAM19322.1 is the non-taster sequence. Under the Coloring pull-down menu, select Frequency-Based Difference. Amino acids that differ between the two sequences will now be highlighted in red. Move the uppermost rectangular slider along the length of the sequence alignment to identify amino acid differences between the two sequences. Record the amino acid positions and sequences.